Yes, perfect. So we are going to start. So today we are welcoming Valery Ozenne from the University of Bordeaux. He will present about cardiac MRI. He has expertise in MRI thermometry and ablation to monitor our temperature change inside of the body. But today he will present another of his expertise related to cardiac microstructure using diffusion MRI and tractography. And he will also mention. He mentioned to me earlier that he has a lot of concept he wants to touch on, but like he will focus the talk on mostly ex vivo for cardiac micro anatomy. And hopefully we will learn a bunch about the first talk that is not about the brain. So since the beginning, I think we had only talk about either the brain or neurons. So I welcome this non-brain talk about the art. So the floor is yours. I will monitor the chat, let people in, and in around 30 minutes, we're going to have a question at the end. If you have any, you can also write it in the chat if you don't want to forget. So Valéry, you can go ahead.
Yeah, thanks a lot, François, for the introduction. Just checking this. Okay. Hi, everybody. Can you hear me? Can you see the presentation?
Yes. Can see the slide and we can hear it.
Cool. Hi, everyone. Thanks for the invitation. So the title of the talk is Tractography as a tool for probing cardiac microstructure. And I'm going to talk about the status, about a few controversies and perspectives. Okay. The plan of the presentation is to briefly talk about myself, just one slide. And then I'm going to divide the presentation into three parts. The first one is to explain what is the cardiac cell structure and how it influences the MRI diffusion signal. And of course, then the next part is to make a short review of cardiac diffusion-based tractography studies and to talk about the associated controversies. And the last part is to talk about some application we're doing in Bordeaux, where we try to combine cardiac diffusion-based tractography and to have a better understanding of cardiac electrophysiology. So this is a large topic, and I cannot cover everything. So the presentation won't cover cardiac anatomy, function, physiology, metabolism. I will really focus on microstructure. I cannot, even if it's really super interesting and it's really an active field of research now, I won't talk about in vivo cardiac diffusion. I will only focus on ex vivo imaging to make the story shorter. And so I won't present cardiac motion, stress, and strain estimation. And lastly, I'm assuming that everybody has a bit of expertise in MRI and diffusion. So I will be really brief on the basis. So about myself, so I'm working in Bordeaux in France. I'm working for the CNRS, is the main research institute in France, and my lab is named the Centre for Magnetic Resonance Biological System. And we are doing MRI development, low, medium, high field. And I have also another lab where I'm working. It's called the Liryc. It's a lab dedicated to the understanding of treatment of cardiac arrhythmia. It's about 30 minutes apart by bike in Bordeaux. And so this is the topic of my talk today. So you can see below a few keywords. And I will try during this presentation to explain interaction, potential equivalence, or what are the differences, challenges when you use tractography in neuroimaging and in cardiac imaging. Of course, I'm not highly specialized in tractography in neuroimaging, but I will try to show when it's possible a few parallels between the field. So don't hesitate to ask questions. So let's start. Okay, so on the left side, you have the well-known microstructure organization in the white matter. And in the heart, we have what we call cardiac muscle cells or cardiomyocyte. So you can see on the right side an individual cardiomyocyte. it has an anisotropic shape. It's about 120 times 20 to 20 micrometer. It can, the shape is changing, so it shorten in the longitudinal axis by 15%, and it increased by diameter of 8% during the contraction and relaxation. And you have to keep in mind the voxel resolution in MRI. So usually for ex vivo diffusion, we can go from 1 millimeter to 100 micrometer, depends on the sample and the MRI. And in vivo, currently for cardiac diffusion, it's about 2 by 2 by 8. So it's really big voxel. So cardiomyocytes are not living together. They form, they are aggregated. So here is a really schematic, simple description of the heart. You have 4 cavity, right atria, right ventricle, left atria, left ventricle. And if you consider the cardiac muscle, so we call it the wall or the myocardium, there is, let's say, 2 definitions. You have the outer part, what we call epicardium, and the inner part in the cavity we call endocardium. And the muscle is made of aggregated cardiomyocytes that you can see on this histological slice or drawing. Okay, so we should not use the term fiber or myofiber. I will use it because it's quite shortened, the cardiomyocyte. But recent review indicates that we should use the term aggregate cardiomyocyte to indicate properties of a collection of cardiomyocyte within an imaging voxel. This is a definition in the last main review of the field. Okay, if you take a perpendicular plane to this image, you're going to see something like that. So can you see the laser, please?
Yes.
Okay, cool. So you have cardiomyocytes are aggregated into sheetlet. So you have 1, 2, 5, or let's say 5 to 6 cardiomyocytes in this direction, plenty of them in the other direction, and then you will have a gap. So it's called the cleavage plane, and it's super important to explain cardiac motion because you have a mix of rotation and contraction, and this cleavage plane is where the sheetlet are sliding. So this is on the right side, another view with confocal microscopy, and you can see the sheetlet here, and here the sheetlet is full of collagen. This is a pathological situation. So it's a non-normal organization. So if you, as a summary, so you have the 2D view with the myocyte. If you take the transverse plane, you can see the sheetlet, the cleavage plane. And if you do a 3D view, you will have this block of myocyte, the cleavage plane. And we can define this pattern like the orthotropic or laminar structure. And you have the main axis, so the fiber, the longitudinal axis. In red, you will have the sheetlet axis, so normal to the fiber, and you have in green the axis normal to the sheetlet. And we are indeed quite lucky because at the scale of the voxel, we have 3 axes that define the arrangements that are perpendicular. So it means we can use SVD or PCA decomposition to characterize this organization. So it's quite nicely designed to use diffusion tensor imaging or, for instance, structure tensor imaging. In this case, the fingerprint will be the cleavage plane. Okay, so if we zoom out a bit, there will be other kind of organization. For instance, in the ventricle, you have from the outer part of the cavity, if you look to the inner part, you will have a rotation of the main orientation of the cardiomyocyte that is linear, and it's about from 60 to minus 60 degrees. This is nicely depicted in this illustration where it's not at scale, of course, where the cylinders, the tube, are representing the cardiomyocyte. So they are perfectly aligned with the circumferential axis in the middle, and then they are rotating again. So it's all result from 1969, and it is called the helical, helix, or rule-based description. There's plenty of paper covering these topics. And then if you zoom out a bit, again, so we move from micro to meso, to macro scale, you have the full organization. And not hard, it is actually quite difficult to understand this scale. As underlined in this recent review, we have plenty of imaging modality to characterize cardiac structure, but still it's challenging to understand what is happening at the macro scale. So ex vivo, we can use MRI, X-ray phase contrast tomography, or standard microscopy, confocal microscopy. So it's really, really close. We're using exactly, I think, the same tools that people are using in neuroimaging. The tool you use will also depend on the size of the sample. So for instance, if you're using a human heart, ex vivo, you will need an MRI with a large bore, and it's super difficult to find a microscopy device that can handle such size. Yeah. Okay, so to end this part, I have an exercise for you. So it's not so easy. So I would like that you focus first on the left screen. So we are looking the tangent plane of the structure. So this is micro CT images from a dehydrated heart sample. It has been imaged at 20 micron, and you can see the myocytes or cardiomyocyte. So if you go through the plane, you can clearly see the rotation of the fiber from something -45, 0 now, and +45. So this is really easy to understand. Now, as a second exercise, you just focus on the middle. So we are looking in a transverse plane, so like the white arrow. And you have, for instance, the easiest part to understand in the middle, the fiber that match the yellow color in this drawing. But if you look in this region, close to the epicardium, you will see some chevron pattern. I'm not sure the exact, I think it works in English, chevron. And now, if you look in the axial plane, it starts to be a real nightmare. So you have to remember, it's really ex vivo, it's static. And when you look, so you have to focus on the right side, the sheetlet are in a way sliding, aggregating, and splitting. And you have a mixed effect of rotation, and it's really difficult to describe and understand. So for us in day-to-day practice, when we have the data, if we look the data this way, it's quite difficult to understand what is really happening at this scale, what's the mean scale. I go fast on that, so we can use, of course, MRI. MRI is super good in encoding the average motion of water molecule in 3D. And if we have some a priori, we can use mathematical model like the tensor to extract matrix that is relevant to describe the microstructural organization. This is a picture of the 9.4 Tesla magnet we're using in Bordeaux. So I go fast on diffusion, but just to show you, we can extract color code FMAP. Oops, the laser. Okay, no, I skip. So orientation is relatively smooth. You have some abrupt change of color in some case. Not able to run the animation. Okay, so I go fast. And I forget to describe it. The big part is the left ventricle. This other part is the right ventricle. And it's image made with only 6 division and 1 biphasm. Okay, so this slide is something I should not show you. This is really critical. If you take any place in the sample and you display the ellipsoid shape of the tensor, so due to the tensor decomposition, you will get a sphere, almost a sphere. So the fractional anisotropy is really, really low. It's about 0.2, 0.4 at best. And so we are using that in day-to-day processing for everyone. So it's, in a way, it's a bit of a disaster, but we cannot change anything. If we add more direction, it won't change. With 6, 35, 35, 64, it will be the same. And if you change the model, so for instance, in this case, I use the model of MRTRIX with the standard 3 compartment used in neuroimaging. I will have a specific FODF. So, and it can be explained by the microstructure because you have the myocyte in this direction, and you have in this direction you have the second main direction is here. And if you look at the histology, there is some let's say oscillation, but in the other direction you have the sheetlet that restrict the diffusion. So I was lucky to be in Sherbrooke some time ago, and Maxime explained to me that I can also take the raw MR signal and apply a radon transform, and we get a similar result. So it's in the data. So in a way, the diffusion tensor model is not fully relevant. I will let Maxime correct me if needed. Okay, so that's all for the cardiac microstructure. I'm going to do a brief historical overview of the use of tractography. It's a selection of some article. I have to apologize because I cannot take everything and present everything. It started, let's say, in 1991 with this paper. Maybe not the first one, but they use microscopy, and they made really nice figure. It's not tractography, but it's look like. And in the beginning of the century, people start to use MRI to scan sample, mostly large mammalian model ex vivo, and they were mostly focusing on one thing. It's extracting the helix or helical inclination angle. So I will explain to you why. What is it exactly? So if you have the first eigenvector using cDTI or diffusion tensor imaging, you can project the eigenvector to the tangent plane, and then you can compute the angle between this projection and the short axis plane. And this will nicely describe the rotation of the myofiber of the cardiomyocyte from the outer part, the epicardium, to the inner part, the endocardium. And then you can plot it a function of the distance, or you can plot the distribution. So it's really important because people are really using in day-to-day this quantitative analysis to explain and compare the microstructure. It's nice because it's quantitative. So in a way, people don't need, not everyone, the tractography. They prefer to rely on this quantitative analysis. But there is some drawback. It's a projection, so you only get a part of the information, and you may lose some patterns. So you can use. There is also other angle related to the sheetlet, but people prefer to work in 2D than in 3D. In the next part, I'm going to present a few of the papers that use tractography. I think the first, maybe one of the first big paper was made by Damien Romer in 2007. So they show nice streamline made in the ventricle. They also reproduce this pattern from endo to epi in the ventricle. They were also really interested to map what is happening at the bottom of the heart, because it's not well described, and you will have a pathway of cardiomyocytes that are coming from the middles and go down and go up. So it's called the apex. And it's really something that is puzzling the community. And on the right, people in these articles, they made also a representation of the sheetlet. And it's quite interesting. And it is important to mention that we don't have in 2026 such computing tools to do that. This is a super important information for us, but we don't have the software, we don't have the tools to extract and display this information. This is something that is lacking in the community. And I find a few papers about what Tractography that people try to apply and demonstrate that some geometric organization of what matter might exist. But in cardiac, it exists, it's for sure, but we don't have the tool to analyze it. Another important paper is a paper from Mortens Murup from the Danish group. So they were really interested to show what are the pathway of the cardiomyocyte. So for instance, they put seed in the papillary muscle. This is some piece of muscle that is not visible here, but that are in the cavity. And they show that the tracks or the streamline are going bottom to the apex, and then they go up and turn around. And it was really an important paper that so behind that, you have to know that there was plenty of theory about the organization of the microstructure. And this paper make a conclusion that is still the actual consensus. So it means that if you put some seed here, you will have a multiple pathway from a unique position, and it's highly reproducible. If I take a sample from my database, if I put a seed in the same position, I will get almost, except it will depend on the contracted state, but I will get almost the same things than us. And they conclude that they have preferential pathway and within the myocyte aggregated. They refute the existence of something is named the unique or unique band. So there is no isolated and easily recognizable tracks. And also the way you see the data, this is super important for neuroimaging people. It should not be interpreted as discrete anatomical bundle, fiber, or tracks. So this is the actual consensus. People, so for instance, this Spanish group that has the other theory. The funny thing is they use the same data and make, or at least similar data, it's diffusion data. They do a similar processing and they get an opposite conclusion. So with the same kind of data, you can defend both theory. Well, in a way, it's a bit funny, but at some point, someone win. And so in 2018, there's two big review by main people in the field, and they say game over to this theory of having a unique myocardial band. So the conclusion are, I go brief on that, but it's ventricular cardiomyocytes are a meshwork. There is no abrupt change, and there is no demarcation. There is no layer or tracks. So it has some impact if you use tractography. This is why I'm speaking about that. And then it was also the end of the game for philosophical question. So you could move on to other topics. And this is what did, for instance, the team of David Sosnovic. I think they are working in Harvard Medical School. And also Professor Choukri Mekawi, it's a French guy. And they work a lot to show that cardiac MR tractography can be used for risk stratification. So for instance, they build a large amount of streamline, and for each streamline, they compute what they call the propagation angle. So it means you take the first eigenvector in a location and you do the scalar product with the previous one, and you will have what I call the curvature of the streamline. And if you have a high curvature, it means that your tissue is infarcted. And in fact, they show it in different spaces, mouse, sheep, human, ex vivo, in vivo. They had really good results. So I highly recommend to check all the work of the team that really push cardiac tractography. And I won't present the paper. I just keep them for the recording, but there are plenty of other paper in other regions like Psychiatria or for making template. Okay, so I'm gonna finish with some application. And before showing some streamline, I need to explain one last thing. So it's about cardiac electrophysiology. It's a bit like the parallel would be the EEG in neuroimaging. The concept is there is a link between the structure and the electrical function. So you all know that for having cardiac beating, you need to activate the contraction. And in the heart, you have a location in the right ventricle. It's called right atria. Sorry, it's called the sinus node that is triggering the electric beating. So it's sending an electric signals that go to the AV node in the ventricle, and then that will go through as an electrical wave in all the heart. And then it triggers a contraction. And it's important to know that the speed of this electrical wave highly depends on the anisotropy of the tissue. So it's twice faster in the longitudinal direction than in this transverse direction, and twice again in the last direction. So it means that you need to understand the structure to understand the electrical function. And we are in a way quite lucky, because if you do ex vivo study, you can take a piece or you can take an heart, you can put it on the table in specific condition, of course, and you can do functional or you can do electrical activation. So it means you can do ex vivo imaging and electrical activation, and you can correlate, for instance, the length or the direction of the main orientation of the myocyte with the function. So in fact, we're quite lucky on that. And it's important, lastly, to understand that this information is not available in vivo. It's mean because there is no sequence that is sufficiently efficient to be used on patient, MRI sequence. So it means people are scanning ex vivo sample, they made model-based cardiac architecture, and then they use this information to perform electrophysiology simulation and to propagate electrical wave. So, and it's widely used for the treatment or the understanding of arrhythmia. And the concept is simple. If you have no structural defect and you initiate an electrical wave, you will have this regular ISO level. But if you have a structural defect, you will have a delay. And this delay will create what we call asynchrony, cardiac asynchrony. In the worst case, it can create really bad arrhythmia, like tachycardic ventricular arrhythmia, like tachycardic ventricular tachycardia. And this is an example of a map recording at the hospital where this region is lately activated. So we try to understand in the lab what is happening, and we try to identify the link between structural heterogeneities and cardiac arrhythmia. And first, and I'm going to try to explain why tractography is really useful for that. So we use sample from human donor. So we have two programs. One is called Harmonica. It's patients that have a clinical disease and they're going to get transplantation. So they usually have a cardiac exam. They get a new heart and we get the old one that is not available, of course, for transplantation. And we have also sample for people having cerebral death. If they have a really bad condition, their heart is not available for transplantation. And in some condition, with agreement of the family, we can get it and scan it. And we are building a large database, so the number is not up to date, in order to improve this understanding. We do MRI. This is the MRI protocol designed by Julie Maga. I'm working with her. She is in charge of all the acquisition. So we do high resolution imaging, diffusion tensor, and like in neuroimaging, we are really interested with collagen imaging. So in neuro, it's for the myelin content. Here it's for the identification of the fibrosis area. And I know that there is some paper from the Sherbrooke team where you are combining watt meter diffusion and myelin-based information using, I think, magnetization transfer or MT or EHMT. And okay, let's start. So we have some nice picture here, but let's continue. The difficulty was to which relevant information can we get from this streamline. So here I use tract density imaging to make the figure. And here is just a big mesh of streamline. It's acquired with 6 direction, B1000, and the resolution is about 600 micron. We are always using this kind of parameter most of the time. And I copy and paste techniques and method from neuroimaging, like streamline description, track selection, filtering, and in order to improve our understanding. So you can see that if you do track selection in up and down, you're going to have some specific area. So this layer, this is the papillary muscle. So it's not everywhere, it's located. And I try to make a relationship between this information that is really coming from tractography and some other knowledge from electrophysiology structure in order to see if we can make some link. So this is the road. So I'm going to do only the 3 point. It's a way to explore the heart. So we're going to start with this region. It's close to the top of the ventricle, and it's called the anterior part. In the literature, you can see in this histological slice that you have something called the ventricular muscle extension. It's some fibers that really align, and it has been reported that there is a high prevalence of arrhythmia in this region, and it's a bit more difficult to cure because if you remove the arrhythmia here, it may continue and extend, and the clinician have difficult to stop it. So we make some images in this region, and we look at so this is a transverse plane. You have the left ventricle, the pulmonary artery, and if you look at the RGB map, you will have something that look like a layer in blue with 2 layers in green. And if you take the transverse plane or the perpendicular view, you will see that a group of cardiomyocytes are aggregated together, and they go in this direction and make a junction with the wall of the pulmonary artery and make a junction with the wall of the aorta. So if you put a seed here and here, or here and here, you will see this nice bundle. And if you put another one, you will get this one. And if you put, for instance, a seed in this region, you will have only 2 pathways. So it was quite interesting to confirm things that have been done on histology, and it was tractography was used for visualization, but it's really helpful to see it in 3D. Second region is about the intraventricular septum. So as mentioned before, it has been described that there is no abrupt change in helical angle in this region. Otherwise, if you have a change of orientation, you will see an abrupt change of the metric. And this is what people were seeing. So we look into the database in all large mammalian species we have, including human. And we found that, of course, in the free wall, so in this part, we have a linear change of the fiber. But if you look in the intraventricular septum, we have something that look like 3 layers, and we have a, let's say, more pronounced change of the helical angle. So we made the cover of AGP and include electrical and mechanical modeling with the team of Ed Wigman. And tractography was really useful to explain what we are seeing. So we can pick a block of tissue, we can display it and show the smooth rotation, and show that here it was not the same in a few, at some point you have an abrupt rotation. And the last one is in the opposite location, so it's called the right ventricular insertion point. It's a posterior, so it's a specific region that has not been really described, because it is a junction between the left ventricle and the right ventricle. And so we look at the data and we show some black signal that was suppressing, but not exactly at the same location on the fractional anisotropy map. So this is in transverse view. We have a reduced value. So the FA was even below 0.2. And if we look at the color code FA, we saw a blue pattern. So it means that the fibers are not going in circumferential direction. They are not rotating. It was not expected at all. And if we look in the perpendicular plane, we can see that it's going from top to bottom. So it was in 2020, I was mostly starting this project. And so I discovered at this time that I can use tools like MRTrix to display the data. And it was also really useful to highlight the size of this block of aggregated cardiomyocyte. And I make a template and show for each of them the kind of the connectivity. Even there is, it's a concept that is difficult to define. And I make some test. I wasn't sure of which parameter I should use to show the figure, and I'm still not sure what is the best stopic criteria that is relevant on cardiology. There is no guideline about that currently. Quickly, it's visible even at people not won't believe MRI alone. So we have to cut the sample, see the triangle, shows it is exactly the same position, and people won't believe the photo. People say maybe it's not cardiac cell, so we have to do histology. And just briefly, the important result is that if you focus on the green square, you are expected that some fiber are going through the plane, through the screen. That is the case here. Then you have a net demarcation where you have fibers or cardiomyocytes that are going in this direction. And then you have again a net demarcation where cardiomyocytes are going through the screen. And we show that it is real and it is in correlation or with some existing arrhythmia and existing clinical phenotype in the literature. So I've finished my presentation. Thanks a lot. I just want to mention that it's just a brief overview of ex vivo imaging, and it's not the life. The heart is beating. This is some animation of Daniel Ennis from Stanford. Maybe one day there will be moving tractography for cardiac application. I don't know. I think it's challenging topics. And as a summary, I think tractography is, I believe it, is really useful to understand the short, medium, or long range arrangement of cardiomyocytes. If you try to have physical question about microstructure organization, it will bring no publication at all because you have a consensus that there is no track, no bundle, no demarcation. Even if you have high resolution data, you will see that it's maybe a bit more complex than 100% no. You can link the microstructure with the function, and it's a small community, a small field of research. We don't have the same maturity that diffusion in neuroimaging. We lack of reproducible code and high quality data. So as it recalled, I'm committed myself to share our existing data set to improve our knowledge. So I would like to thank all my collaborators, especially Julie and Pierre, my former PhD student, and all the people from the Liryc and the imaging team, and of course the community.
Bravo.
Thank you very much. That was a great talk, a very good overview of so many things I didn't know about the heart, and also how tractography can be used. So that was very nice. We're gonna proceed with questions. So like if there is anyone with question, you can raise your hand, you can unmute yourself and talk. You can also put it in the chat if you want us to cover it. To break that last, I will ask a question about more context for clinical cardiac MRI, because I know nothing about this field. Like in the day-to-day life of a clinician for cardiac MRI, what's the kind of acquisition? Like they do something just to look at the movement. Like I assume they don't look at microstructure because like since it's moving, I assume they look at the slice that move in real time, and this is informative. So how does those acquisitions perform in clinical?
It's really an active field, so there's lots of things happening, but basically the standard exam include mostly 2D images. It's always 2D. So you're gonna check what we call cine images, as you suggest, is to check the motion of the wall and to be sure that you don't have asynchrony. Then the second thing is to do flow imaging to be sure that you don't have regurgitation, that the blood is really coming from the atria to the ventricle and not coming back from the ventricle to the atria if the valve are damaged. There is some perfusion imaging to be sure that the tissue, the cardiac tissue, is perfused. And lastly, people are really looking at the viability of the myocardium, but they are using a contrast agent, gadolinium-based imaging. And the diffusion community is trying to push the sequence to use diffusion and avoid the use of contrast agent to assess the viability of the myocardium. But it's in progress, but it's absolutely not something that is done at the clinical scale. It's only done in research center, but it's working. In the presentation, I put some review, and if you're interested to see some case, so the team of London in UK, in US, there are a few teams that are really pushing new sequences to use diffusion and to characterize the viability of the myocardium.
Thank you for the great answer. I see Maxime as a raised hand. Maxime, go ahead.
Yeah, thanks, Valery. That was amazing. And I think we can publicize this talk as being a great historical overview also for people getting in the field. So thanks for making that work for us and not, being too biased by your own work. This was really nice. Very, very nice. As you may know, we're currently working on the grand challenges of tractography. And we have a long list of challenges, and it's heavily biased towards the brain and neuro. And you kind of gave us a nice indications of where you kind of tweaked the existing software for your needs. But what would be your top 3, let's say, challenge for our toolboxes Because from what I see here, it's very supervised tractography with manual seeding, manual termination. So you're kind of tracing the road in your Google map that you're looking for. But how do you go towards more like unsupervised, automatic analysis of these structures? And what's your top 3 challenge?
I think the first one, the most important, is to be able to show the fiber and the sheetlet organization. The paper for 2007 from Damien Romer was really great. It's never been really, there are few attempts to reproduce it, but we don't have software where we can see both both organizations together and also at different scale. So it's like sometimes you want to work at the scale of the voxel, and sometimes you need to work at maybe at the scale of 1 or 2 centimeters to really understand what is happening. So this is the let's say my first wish. And the second one, it maybe would be really, I build once a connectome, so I divide the heart into region, and then I made the I don't know the name. So when you do a connectome, you have a map, a circular map.
Yeah.
And so I do it for the heart, and I try to see the connection in the region. It was really nice to do because it's making nice figure, but I didn't find a way to push it. But it would be interesting also to have, let's say, a standardized connectome of pathway. I don't know for what, but at least for the understanding. And
So where would you place these regions?
there is some existing division, but it's completely arbitrary. There is arbitrary rule in the field. But yeah, where do you place this region is start to be complicated, and some funny things to do. Maybe but it's not so it's not about science and pathology. It's about visualization. For instance, so you have the right ventricles, you have the left ventricle, and if you take really high quality data at the junction, you will have something like that. You will have plenty of fiber doing this, so you can see it's super easy to see on the data, but it's a nightmare to display. And if you want a big challenge, I can share the data. And if you can show en français, je dirais ce tricotage, mais je ne sais pas comment on le dit en anglais, si tu peux m'aider.
When you knit this, what's it called in English? Like a wool knitting.
It's interweaved.
Yeah, interweaved.
Yeah. And the interesting thing is this feature is visible in MRI at 600 microns. So I mean, it's done on a scale that is visible on the data. It's not at the lower scale. You can see the fiber and bundle. If you want a challenge to display that, it will be really nice.
Thanks, Valéry. I don't want to put anybody on the spot, but I'm really curious to hear Kathy Rockland, who's been following brain axons for 50 years to comment on cardiac cells. And do you see parallel or things that can?
Yes, I do. I wasn't sure I would make the full talk, but this has been marvelous. It's extremely exciting. I'm actually listening to the issue of not single axons, excuse me, very exciting, but fasciculation. Fasciculation occurs. If you remember my talk at Sherbrooke, I can send you a little bit later the fasciculation after an injection site. But, and it's basically the probably optic radiations that's too large. But if we did a fine fascicle analysis of a defined fornix, perhaps, corpus callosum, you will see fascicles. And I'm wondering, not only do they exist as structures, but is it related to waves, brain waves, and conduction velocity? So I think this is right up there with millennial challenge. You're registering. Okay, I'm not conveying,
but
but
yeah, it's really for us. It's really nice. We can have association between structure and conduction velocity. That's something that is really interesting for us. We can bring information for other field. If we use tractography alone, we cannot make conclusion. It's really in support of what we are seeing in for instance, in cardiac case for conduction velocity of electrical wave.
Yeah, I mean, what I said could be Thai blue, sky blue, but to I mean, for the whole talk, I was just seeing yes, yes, yes. I bet it's true in the brain. So it's great.
Thanks, Kathy.
I see a question from Kurt in the chat. A question that I had myself about what kind of constraint we can use in tractography when we initialize tracking. Like one very famous tractography constraint in the brain is like the stopping criteria. Like when you reach nuclei, like we know that axons have a start and a beginning. Like they are not like infinite. I think that's not the case for the heart. Like technically you can loop around because it's the muscle fiber just touch itself at the end and it's a continuous element. So do you just put a length threshold for visualization purposes? Like you just wrap once or twice and then you stop it? How do you define that?
It's an interesting and difficult question because there is no consensus on that first. The only consensus is it's going to wrap continuously most of the time because we, in agreement with the consensus, it's a continuous mesh or an aggregate mesh, so there is no demarcation. But in practice, when you look the data, there is some region where it stop and start because you have the junction with the atria. You will have a few places, but it's much more an exception where it stop. But it's much more it's an exception to the rule. So there is no in general there is no start and stop. But there is exception to the rule, I think so. I will say like that. And anatomical constraint.
Do you have a concept of like, because sometimes in the brain we have like axon diameter and we kind of hope that in the future we can say the axon diameter along a group of streamlines should be stable or like continuous or something to say like you shouldn't have a small axon diameter and then jump to a gigantic axon diameter. Is there something similar for
the anatomical constraint? The anatomical constraint should be the sheetlet organization. So it should be related to that. So if you have, because you have, I didn't talk about that, but there is also a way there is some metric about the sheetlet inclination and things like that. So it should be related to this. And the only constraint you have is the vessel. There's plenty of vessel. If you look, there is a few constraints in anatomical constraint, but it's the existence of vessel in the heart. And it's at some point it's a question of semantic. And my level at some point I reached my level of English, and it's really difficult for me to discuss about the semantic because when you read the paper, it starts to be semantic, philosophical when you talk about the limit. But maybe there is work to do, and maybe we can really use tractography and to extract quantitative metric as you do in neuroimaging. Maybe there's something to do also.
Thank you for the answer. I see Alexander as a raised hand, so you can go ahead.
Yeah, so first of all, thanks, Valery, for the nice talk. I had a question about the architectural configuration of these cardiomyocytes being organized in these, laminar sheets, right? By the way, quick intermezzo, Chantal Tax and I, together with Kurt Schilling, we're implementing the SPI metric, so it will become available soon. That's a separate
Cool.
separate note. So yeah, we're kind of reviving it again. Also partly based on Tim Derby's revival of the sheetiness of the brain and stuff like that. But that was not my question or my comment to begin with. I wanted to say something about the fiber ODFs that you also reconstructed, right? So there's no real crossings in the heart configuration of the tissue, right? Or is there?
No, that's a good question. I have prepared a slide, but I think I can just answer like that. So there is no figure crossing, but due to the partial volume effect,
Yeah. So
it's
due to the
region
size, the
where
finite size of the voxel.
yeah, we for instance, I show the triangle, and if you do FODF reconstruction close to the triangle, as you have fibers are this way or this way, you will have ODF with two directions, and we can use NUFO, the NUFO metric, to quantify where the partial volume effect happens. It's about,
Yeah.
but it's about 1 or 2% of the volume, so it's not a lot.
Yeah. So one thing I was thinking of is that if you've got that prior knowledge of that partial voluming, for instance, going outward, inward, or even up, down for that matter, wouldn't it be possible to use that as constraints in the estimation? Like you use that to kind of come up with a, I don't know, cardiomyocyte OD instead of the FOD, right?
Yeah.
So like a
Yeah,
COD,
It's a really good comment. We had this discussion with Maxime, and when I did the FODF reconstruction, I used the axon model. It makes no sense. It's not absolutely. It's absolutely wrong. Indeed, it's not completely wrong.
Exactly.
So a model is a big deal.
Yeah. So one of the things I was thinking of is that obviously with FODs and think of the brain stuff, we always have like a nice axially symmetric response function, but I would just design the response function to be a sheetlet like a uni-sheet function. You know what I mean? Like you basically deconvolved with a sheetlet instead of a fiber ODF, right? Like and because you've got prior knowledge about the configuration like inward, outward, upward, downward, you can actually use neighboring voxels to give you like a recursive updating of your sheetlet at that location to use for your deconvolution. So you would do a voxel-wise response function estimation based on your sheetlets, like uni-sheetlet structure, and then you would end up with your degrees of freedom that you're interested in, right? So you may actually come up with an asymmetric, COD. I'll call it what it is now, then, right?
I don't have the skills to do that, but I totally agree that this is something that should be done. Yeah.
Anyway, something to maybe to team up with. I'm looking at the technical people now. I see Francois, Maxime nodding, so maybe we can have a student working on that. But anyway, it's just a thought.
Yeah, it's fun. Thanks.
I don't see any new question in the chat or raised them, so I think on that we can conclude. I see that Kurt is saying not a question, but from nice but useless presentation is favorite slide title. So I agree, that's true. That was funny when you showed it. So I think we can conclude here. I'm gonna stop the recording.
Thanks a lot, Francois. Thanks a lot, everybody, for the discussion.